Let's meet the Charles Sturt team who study the strange, unusual, weird, wacky and wonderful world of parasites. But what is a parasite? No, it's not your friend or sibling who sleeps on the couch and eats all the food in the fridge, although the principles are similar. Let's ask an expert, Dr Drs Shamsi.
First, let me tell you what the word parasite comes from. You may be interested in knowing that this word is coming from a Greek word parasitos, which made up two different words. Para meaning neighbors or friend, and sitos meaning bread, food or grain. And basically in old Greek times, they used to refer to people, neighbors or friends that live on other people's expense. For example, they go to their house when it's only close to the dinner time as parasitos. Later scientists use this term to refer to creatures that live on the expense of other creatures, known as hosts. So as you can imagine, parasites can be quite harmful at times because they rely on a lot of food from their host to live on.
How and why did you become fascinated by parasites?
I was always interested in science and how our magnificent natural world works. When I was an undergraduate biology student, I became very interested in taxonomy. And taxonomy is not really just a big fancy word. It's the science of how to classify and organize living organisms, because we are lucky to live on a planet that is full of diversity and we have many living organisms living there, and it's fantastic to have, and as we know so far, it's a really unique thing on the entire universe. So for me, taxonomy is not a boring field of science as some may think, because to classify things and to be able to organize them into different groups, you actually have to learn about so many different aspects of the organism you're working on its taxonomy. You have to know about its anatomy, you need to know about its DNA these days, you need to know about its behavior and also about all different aspects of its ecology to be able to properly assign it to the right group. So when it came to parasites and parasitology, through taxonomy of parasites, I started to learn about so many aspects of these fantastic creatures. And I have investigated actually the deepest layers of nature, animals, plants, and humans through their parasites.
Why is this study of parasites important?
The main reason is parasites are the biggest group of creatures on Earth. In other words, there are more of them than there are of us or any other creatures for that matter. So by numbers, parasites rule. No one should really ignore more than half of the Earth's biodiversity, but unfortunately, commonly it happens, and a lot of us ignore their presence. The main reason is most of them are really small creatures, and we need microscope to see them. Other reasons that these parasites are so important and we should know about them and study them is that they are among the deadliest creatures for humans. And in Australia especially, parasites cause the highest income loss to farms and stop farms producing as much as they should.
Now I'm confused. I thought we were here for a drawing lesson. What's that got to do with the study of parasites?
When we talk about the art of drawing, we might automatically think of a master like Leonardo da Vinci, or drawings in our comic books. But scientists do beautiful drawings as well, like this one which are parasites found in Australian sharks. A parasite portrait, as you can see in this picture, is an amazing line drawing. The details are really incredible, don't you think? So next time you hear the word scientist, think of some of us as great artists as well. Unfortunately, there are not many parasite artists or taxonomy experts anymore, and that's why one of my missions is to train the future generation of taxonomists to have this skill. Now let me take you back to the lab and introduce you to some of my colleagues and show you fantastic works they do.
Now I would like to introduce you to my colleague, Dr. Drs Barton, who, as you can see, is very busy. Hi, Di.
Hello, how are we?
Good, how are you?
Oh, I'm very well. I'm looking down a microscope.
Yeah, I was going to ask what kept you busy today.
Yes, I'm looking at pentostomes.
Ah.
Pentostomes are parasites primarily of reptiles, but also of some mammals, and they live in the lungs of reptiles or up the nose of mammals. So.
Fantastic. How do you get hold of them?
Unfortunately, the animal has to be dead when you cut it open, and with the reptiles, you just dissect them open up the lung. A reptile lung is like a big sack, so it's nice and easy to get into, but with the nose, you have to then cut the head in half to get right up into the nose to be able to pull them out.
Are they really bad for their host?
They can be. They are blood feeders, and they have hooks that they use to grab on to the wall of the lung or the nose, so they can create a bit of a problem.
How did you become interested in them?
Oh, they were just so awesome to look at.
All right.
Okay, to work on parasites, we need to see what's happening inside the parasite itself. So we need to look at their internal organs, which is quite important. Because they're so tiny, it's hard to do dissections on them. So we put them on slides, look at them down a microscope, and then we draw what's happening inside. So what we have here is a microscope with a slide with a specimen on it, and we have this thing coming out the side which is called a drawing tube. And so this is what we use to be able to draw the specimens, because it's very hard to do a freehand drawing of a parasite to scale. We need to make sure it is to scale when we do the drawing. So basically when you look down the microscope, you can see the specimen down under here, but then over here you can actually see your hand underneath this drawing tube. And so it's basically tracing the specimen. So what we'll do is, let's turn down the light so I can see it, is we draw the specimen, and the specimen is now being drawn to scale. It's got an intestine tube inside that we can see coming up like this. And then it's got little lines coming across on the outside like this that we can also see. So these are things that we can now use to try and identify the insides of a parasite. So it takes quite a while to do a really good drawing to get all the details. You can have lots of different parts of the parasite. So what we have here is a picture. This is a nematode, not a pentastome that we've drawn earlier, and this is the level of detail that we can get out of these drawings.
So can you please explain what we are looking at in this nematode?
Absolutely. So I'll just grab my pencil here. So this is the mouth of the nematode here and the lips that surround. So nematodes have different parts around their mouth. They have different structures to their lips and little structures there. These things are called phasmids or papillae. And then we have cuticular inflations here coming down the outside. And then you've got the esophagus coming down through here. So it shows all the internal structures that we can then use to determine all the different species, doing measurements and seeing if there's differences in the patterns. With the photos is they are truly just two-dimensional, so you can only ever get that one focus plane. But with a drawing, you can focus up and down and you can actually draw things in the way that they are, so you can put things on top of other things. So it just makes it a more accurate representation. But just to show you again with the drawing, I've just turned on the lamp here and you can actually see my hand coming down through here. So this is how we trace. You get a bit of a lag because of the video, but you can use this to trace, and it's hard because it's now backwards, but you can trace I'm going the wrong way, there we go, the things there with the pencil. So that's what I see when I look down the microscope, is my pencil down on the microscope there, and so that's how I do my drawings.
Hi everyone, I'm Dr. Shamsi, also known as Diego. I'm from Charleston University. Today I'm going to show you the parasites that I'm working on. The parasites that I'm working on is called Lania species. One of the most famous member of this genus is probably Lania crabiensis, also known as ankleworm. It's widely distributed around the world and probably introduced to Australia through carp or goldfish. It caused significant loss on freshwater agriculture and then impacts on freshwater fish welfare. It has been now found in many Australian native fish species, but at the moment we don't know if all those linear species found in Australia are belong to the same species or not. And also, do we actually have any Australian native linear species here present? This slide shows my drawing of these parasites. On the left are the holdfast, or the ankle of this parasite. It's the part that insert into fish tissue. And then on the right are the leg of this parasite. So these ankles are the single most important taxonomy features for species identification in this group of parasites. But here you can see from this drawing, the morphology of these ankles change significantly, depends on the host species and then where these parasites attached to.
Thanks to the Charles Sturt parasite team for this fascinating insight